for coming today. It is a pleasure and an honor to introduce friend and colleague James Brown, who is going to give 2 talks today. He's going to give our departmental seminar now, and at 4 o'clock he's going to speak at our research symposium, which is going to be held at the Sierra room, is it?
Sequoia room.
At the faculty center. Please come earlier and look at the posters that are there as well. Jim got his bachelor's degree AD at Cornell and PhD at Michigan. He postdoc here with Bart and then was hired as an assistant professor before he continued his life as an academic nomad and traveled around to
Utah, Arizona, New
Mexico, and now he's currently at New Mexico and also at the Mechanical Institute. I'm going to tell my fun stories about Jim later, so you got to come there. I'll repeat this one because it's a good one. He's an amazing, theoretical ecologist, but also an empirical ecologist, and probably the most renowned analyst living. Highly, many awards. But what really excites me about Jim is his really good eye for natural history. So I work in the Rocky Mountain Biological Lab in the summer, and Jim was out giving a series of distinguished lectures, and I'm walking around every day, and so I was noticing some beavers were talking about really big trees up a really steep hill, 80, 100 meters away from the water. And I'm walking by this every day, and then Jim comes into lunch carrying these discs, these wooden discs. Beavers create the wheel. They were cutting this tree into pieces and rolling the discs down the water. So what's that about? I've never seen that before. He's never seen that before. No one at Rumble had ever seen that before. And he's just there visiting and was teaching us all about natural history and so in addition to being an astute natural historian, because he's such an astute natural historian, he's also an amazing theoretical ecologist, and today he's going to share with us toward a metabolic theory. Please join me in welcoming Jim.
Thanks. Let me put this back on here. It's great to come back. Have wonderful memories of my time here. And yeah, Dan, so far as I know, that's still in a hypothesis. Those are my glasses? No? I've got mine. We should have gathered some data on those beavers, unless you did, to make sure that's what they're doing. But anyway, it was a fun time we spent there. All right, so what I'm going to talk about now is this stuff called metabolic ecology that has sort of dominated my research interests for about the last decade or so. And I guess I'll just jump right in. So the first point that I want to make is why look to metabolism as a mechanistic and theoretical basis for ecology. And I often introduce this by saying that metabolism is to ecology as genetics is to evolution. Here's why. What is metabolism? It's the flux and transformation of energy and materials in organisms. And this involves really 3 components: exchange with the environment, processing within the organism, which is metabolism in the narrow sense that a biochemist or a physiologist would emphasize, and then allocation of energy and materials to fitness, to some combination of survival, growth, and reproduction. What's ecology? Ecology is interactions between organisms and their environments, both biotic and abiotic. And what is an interaction? Every interaction involves an exchange. If you think about it, an exchange of energy or materials. It can be information, but it still has some energetic or material form. So in this sense, most ecological processes are fundamentally metabolic, and we can look to the rules that govern those exchanges for the mechanisms that underlie a great deal of ecology. So we've been developing this thing that we call a metabolic theory of ecology, and I'd sort of like to give you 2 flavors of that to start this talk off with. One is what I would call the sort of soft version, which is metabolic theory of ecology in lowercase. And here the idea is pretty much just what I've articulated. There are general linkages between ecology and metabolism, and they involve both energetics and materials in stoichiometry. And this version of the theory would assume that, for example, that metabolic rates and ecological rates vary with the attributes of organisms and their environments that govern metabolic rate, that is with body size, temperature, and stoichiometry, but we don't necessarily have to assume any particular mathematical form or parameter values. So in this sense, it maybe is like evolutionary genetics as just a big broad topic. Then there's what we might call hard, or MTE as it's been termed by others, and we've adopted this now, the metabolic theory of ecology, which is developed particularly in this paper that we published in 2004, which I'll develop in just a minute. And this is the more specific theory. And so this does imply particular equations and sometimes parameter values. And so, you know, to continue this analogy with genetics being the basis of evolution, this might be more like Hardy-Weinberg or Fisher's sex ratio theory for ecology. And so what I'm going to do is present this rather hard version with the sort of caveat that in many cases I'm willing to back away from that a bit if it doesn't work, but I'm pretty much unwilling to give up the soft version, that most ecology really is about metabolism. So here's the hard version, that metabolic rate, mass-specific metabolic rate, and most ecological rates scale with body mass and temperature, and what I've tried to do in most of what follows is keep the body mass term in blue and the temperature dependent term in red, just to make it easier to follow through. So a rate like metabolic rate or the intrinsic rate of increase of a population scales with body mass and temperature with these three terms. One is this normalization constant that sits out front, and that's a constant as far as the math is concerned, but the value of this constant can vary with taxon and with environmental conditions, for example. The mass dependence we take as a power function with normally what we call this quarter power exponent. So as mass to the minus a quarter power. And the temperature dependence is this exponential term here given by what's called this Boltzmann or Arrhenius factor, where E is the root of the natural log, and this is raised to the power minus E over KT. So K is Boltzmann's constant, T is temperature in Kelvin, and the temperature dependence is given by the value of E, which we suggest for most processes that are governed primarily by aerobic respiration, the value of E should be about 0.65 electron volts. And what we can then do is take those equations and simply rearrange them. The equations almost tell you how to plot them to analyze both the temperature dependence and the mass dependence. So if you plot the logarithm of the rate and you adjust for the mass dependence, either by putting in a mass to now plus a quarter because it's on the other side of the equation, or you can put in the observed mass dependence. And you plot that as a function of 1 over KT. This is what's called an Arrhenius plot. And the prediction is that that relationship will be linear, and the slope will be minus E, and where E, if it's, again, the process dominated by aerobic respiration, will be about minus 0.65 EV. If you like to think in terms of Q10s, this means that the Q10 would be about 2.5, a typical biological Q10. Similarly, you can do the same thing for the mass dependence. So you plot the logarithm of the rate, now temperature adjusted, versus the logarithm of mass. And the prediction is that'll be linear, and the slope will be minus a quarter for many processes. And so this just is, shows you proof of concept, as it were. So what we're looking at here is development rate of zooplankton eggs measured in the laboratory. And we're going to look at temperature dependence first and then mass dependence second. So you plot the logarithm of the hatching rate, mass adjusted. So here we've done it theoretically using body mass to the quarter. And you plot that as a function of 1 over kT. So this is inverse temperature. The cold temperatures are on this side. The warm ones are up here. And these are experiments in the laboratory with various rotifers and cladocerans and whatever the other guys are. I can't remember right now. So these are experiments in the laboratory, and they're conducted, as you might expect, at 5 degrees C intervals. And here's what the data for a wide variety of these organisms look like. Here's the regression line fitted to that. And the prediction is that the slope of this should be about minus 0.65, this with the sign reversed. And you can see it's a little bit steeper than that. And temperature alone, well, mass-corrected temperature accounts for 90% of the variation. You can do it, take exactly the same data from the same experiments and measure the mass dependence. And in this case, we're measuring the egg mass, which is actually the mass of these offspring when they hatch out. So here's the hatching rate, temperature corrected, and plotted as a function of egg mass. The prediction is the slope of this relationship should be minus a quarter. And you can see it's very close. And we're accounting for about 85% of the variation. Now, the other thing you can do, once you have this theory, which doesn't account, obviously, for all the variation, is you can say, well, does this theory help us explain some of the residual variation? And so what we're going to do here, we've accounted for 90% of the variation in hatching rate with temperature. And now what we're going to do is examine one hypothesis for the residual variation around this line. And that hypothesis has to do with stoichiometry in a large body of work on ecological stoichiometry. Sterner and Elser have suggested that particularly important in governing the rates, especially of freshwater aquatic organisms, is phosphorus, because phosphorus is often limiting in freshwater systems. And so what we're doing here is we're plotting the deviations around that temperature relationship as a function of the carbon to phosphorus ratio, right? And so high body phosphorus is hypothesized by Sterner and Elser to give rise to rapid developmental rates, low to low rates. And you can see that remember, we accounted for 90% of the variation before, and we account for 2/3 of that remaining variation with body composition measured as C to P ratio. And so this just gives you, this is done then to give you some idea of the foundations of this theory and how we apply it. Now what I want to do is just present kind of a smorgasbord of a series of topics that we've investigated from this metabolic perspective, which interest me at least, and hopefully some of them will interest you. So the first one has to do with a scaling of metabolic rate and ecological rates across what we call the early transitions of life. And the idea here is, this is highly oversimplified, of course, but the idea is that the first organisms in the world were prokaryotes, and they were very small. Then we had the symbiotic mutualistic origin of the unicellular eukaryotes, so protozoans and unicellular algae. And then these organisms remained together and formed little colonies, and evolutionarily eventually gave rise to multicellular plants and animals. And so the old dogma was that the scaling relations that we see in metazoans and large complex plants and animals just carry right on down to these very simplest organisms. And what we did, and by we in this case I mean in a project led by 2 of our graduate students, and I don't think I've ever done anything myself in the last 20 years probably. Almost everything I've done has been collaborative, and a lot of the inspiration has come from students. What we did was gather information from the literature on unicellular eukaryotes. We focused on heterotrophic organisms where respiration was the primary mode of energy production, and we measured respiration rate or metabolic rate, the maximum population growth rate, little r max, and production efficiency. That is, how much growth does one get per unit energy consumed. So here's what we thought we were going to see. This is comes from a very large synthetic study by Hemmingson in the 1960s, and this is kind of where we were starting our study of the scaling of metabolic rate. So here's metabolic rate over here. Here's body mass here. These are logarithmic scales. We're now measuring whole organism metabolic rate. So we expect if the per unit mass metabolic rate scales as -1/4, the whole organism metabolic rate scales as 3/4. So, for every 4 orders of magnitude increase in body mass, metabolic rate increases by 3 orders of magnitude. And Hemmingson had plotted out the data for what he called homeotherms. We now call these endotherms, poikilotherms, now ectotherms. And he plotted separately unicellular organisms. And you'll see that he fitted parallel lines to these. He fitted slopes of exactly 3/4, and the difference in the elevation is the difference in that normalization constant. So the main thing is that he suggested that this 3/4 power scaling extended from these multicellular organisms right on down through the unicellular ones. So what happens if you gather the data? You get a very different picture. And this is, I think the most accurate representation that we could get from given the current literature. So here are the metazoan organisms here, and their slope is 0.82, and we maybe want to come back to that a bit, but their slope is definitely less than 1, significantly less than 1. Here the unicellular eukaryotes, protists, and the slope is very close to linear, very close to 1. The confidence intervals certainly include 1, as you can see here. And here's the slope for the prokaryotes, the heterotrophic bacteria, and their slope is almost 2. So very different than the original literature would have led us to suggest. If you plot here mass-specific metabolic rate, and let me get clear which one this is. Yeah, okay, this is mass-specific metabolic rate here. So we're dividing by the mass of the organism. Then the prediction is this slope would be about -1/4 all the way through if it holds for everything. Here's what you see with those linear slopes for the eukaryotes, unicellular eukaryotes. The slope then is virtually flat, and we still get a slope of about 1 for the bacteria. And if we then measure one of these ecological rates, little r max, the maximum rate of population increase, we find that the slope for the metazoans are pretty parallel. The slopes for the prokaryotes are fairly parallel. And there's this interesting phenomenon, though, that suggests that the unicellular eukaryotes have a negative relationship between population growth rate and cell size or cell volume. And then this is what happens if you measure this production efficiency, which I think is kind of an interesting phenomenon. So this is how many grams of biomass are produced by growing populations per joule of energy consumed. And what you see is that there don't seem to be any significant size dependencies on this in each of these three groups, but that the bacteria are significantly more efficient on average, although there's a lot of overlap. The bacteria are more efficient than the unicellular eukaryotes, and these protozoans are substantially, almost an order of magnitude more efficient than multicellular organisms. So how do we explain this, and particularly how might we explain this interesting this very steep scaling of metabolic rate in the prokaryotes? And the basic answer is I don't know, but this is kind of intriguing, that there is in these prokaryotes, in these bacteria and a few archaea, there is a pretty close scaling relationship between genome size, or the number of genes, and the mass or volume of the organism. And so if in some sense the metabolic rate, the ability to generate power, depends on the size and complexity of the genome, this at least seems to provide a testable hypothesis for the steep scaling that we see in the prokaryotes, in the bacteria. So, I'm going through these pretty fast. So it looks like, in this case, that the old dogma is wrong. The exponent is not about 3/4 in unicellular organisms. In prokaryotes, it's close to 2, and maybe this reflects scaling of genome size, at least in part. In unicellular eukaryotes, the exponent is close to 1, and maybe in these heterotrophic organisms, that simply reflects linear scaling of mitochondria. And Jordan Oakey, one of the co-authors of this, has worked on this. In metazoan organisms like mammals, the fraction of the body mass that's liver or lung or heart, most organs, scales linearly with mass. We have the same fraction of those organs. And so how many mitochondria can you fit in a unicellular, in a protist? It looks empirically as if the data support this, that not so much the number of mitochondria, but the total volume of mitochondria, or total area of inner mitochondrial membrane where the energy transformation actually occurs, scales roughly linearly with body size in protists. And in metazoans, and we'll look at this a little bit more, a similar case as we go from solitary organisms to eusocial organisms. The hypothesis here actually is that the scaling relationship changes. It goes from 1 to approaching probably something close to 3/4 with increasing body size. Initially, when we have metazoans that contain just a few cells, the metabolic rate is simply the sum of the metabolic rate of those cells, and it goes up linearly. But as you build a larger and larger, more complex body, our theory for the scaling suggests that then you have to supply that body with nutrients. You have to carry away the waste. You have to do the biomechanical support. And you run into these diminishing returns, which enforce sublinear scaling, an exponent less than 1. And we suggest that approaching this sort of canonical value of 3/4. So that this is owing then to the constraints of resource supply and vascular systems. And the production efficiency is much higher in prokaryotes. All right, so let's look along at another one of these evolutionary transitions, which is as we go from individuals to societies and social insects. And there are 2 different groups that have worked on this. One is Chen Hao and Jamie Galuli and Mike Cosbury, all people that have collaborated quite a lot with us. They compiled data from the literature. So these are metabolic rates of solitary insects. So these are just insects, individual beetles at this end, and little tiny collembolans or whatever at this end. And then here we have data from a number of insect colonies And I think these are all Hymenoptera. They're either ants or wasps and bees. And they fitted slopes to both of these relationships. And the implication here is that these are parallel. And they then suggest that to think about this sort of in metabolic terms, yeah, an insect colony in terms of its metabolic requirements and how they scale with its size really is a superorganism. And the scaling with mass is relatively similar. And there is here in their data a hint of a significant displacement, a change in that normalization constant. So it takes more energy on average, adjusting for size, to support a colony of insects than a social insect. And that's something that, in fact, we're quite interested in and beginning to work on. Here, even more dramatic data measured experimentally by James Waters and John Harrison at Arizona State. And they worked on ants. Here's 7 species of individual ants and their metabolic rates measured. And so this is log of metabolic rate against log of mass. They temperature adjusted them, and the slopes of these vary from 0.55 to 0.83. The average is almost exactly 3/4. And then they measured entire colonies of 2 species of ants, Pogonomyrmex californicus and Phytoli dentata. These are common desert ants, if you know them. And what you see is that they weren't, I don't think they still are, devotees of the hard version of metabolic ecology, but the slopes of both of these relationships are very, very close to 3/4. And what you see if you extrapolate them back is that there again is this displacement in the normalization function. And it's interesting, at least, to think about the hypothesis that the greater elevation in Phytoli dentata is because there's a higher degree of social organization in Phytoli than there is in Pogonomyrmex. We know that there are distinct segregated morphological castes, soldiers and minor and major workers in Phytoli. There are not in Pogonomyrmex. Phytoli, if you just watch them in the field, they seem to forage on a much more organized trails and so forth. And so it's interesting to hypothesize that this higher degree of social organization requires more energy expenditure. It's like the second law of thermodynamics. You have to put more energy in to maintain a more complex system farther from thermodynamic equilibrium. So the bottom line here is that the metabolic rate of individual insects seems to scale close to mass to the 3/4. The metabolic rate of entire colonies also appears to scale close to mass to the 3/4. And if you look, here, in order to get from here, if these are Pogonomyrmex californicus to here, as colony size increases from 1 ant to this size, there has to be a steeper slope in here. And again, the hypothesis would be that there's a roughly linear portion here when the colony is very small, and then as it gets large, it begins to develop complex networks of resource acquisition and supply and so forth. And as I say, we're working on that. So this suggests that there is an economy of scale which may be in response to resource distribution and resource acquisition and distribution, similar to that we see in whole organism metabolic rate. So we might now sort of step back and ask, well, what good is this stuff? Does it say anything about some of the problems of the world? And I should mention, I'm really excited, but I decided not to put it into this talk. But I'm working right now in collaboration with Chinese investigators, and my Chinese collaborator is the director of an agricultural station in China, northwest of Beijing, and we're doing quite a bit of work on the application of metabolic theory to crop plants, and whether we can, and we're asking the question, can we develop models that, for example, predict the optimal density to plant seeds to maximize yields of different crop plants. And if we get a chance to talk individually or whatever, I'd be delighted to talk about some of that. But let's focus in for the moment on the scaling of the carbon cycle. And so how does carbon turnover in ecosystems vary, first of all, with plant size, and secondly, with temperature? So originally, I and some of my collaborators got together some of these data, and now more recently, Krista Anderson-Teixeira and Peter Vitusek, and this actually is no longer in press, it's just come out in our Metabolic Ecology book, have gathered additional data and performed a new analysis. So here is the scaling of ecosystem carbon turnover rates as a function of the size of the dominant plants in those ecosystems, right? So we have forests at this end with these black dots, and we've got aquatic ecosystems with phytoplankton over here. You know, we've got 10 to the minus 15 to 10 to the 8 or something like that. So we've got something like 16, 17 orders of magnitude of variation in plant size. And the prediction, you know, the sort of naive prediction would be that the slope of this relationship should be minus a quarter. And you can see that it's not too bad. It's minus 0.21. And, you know, obviously these systems with small plants are turning their carbon over very rapidly. And in forests, it's turning over much, much more slowly. And, you know, I think one of the messages from this is, well, if I want to grow plants for biofuels, right, I grow algae, not trees. And if you remember the temperature dependence, I grow those algae in Phoenix, not in Fairbanks, right? And so now let's look at the temperature dependence. And there's an interesting phenomenon here that I haven't told you about, but which has interesting implications for the anthropogenic global warming, this huge insult that we're imposing on our planet, for the carbon cycle. And the reason for this is that photosynthesis and respiration have different temperature dependence. I told you that measured in electron volts, the temperature dependence of respiration, aerobic respiration, is about 0.65 electron volts. So the slope of that relationship is roughly minus 0.65. The temperature dependence of photosynthesis is about half of that. It's about 0.35 electron volts. And you know, this has been pretty well worked out by the biochemists and plant physiologists. So the slope for photosynthesis is going to be less than the slope for respiration. So in terms of thinking about the global carbon cycle, we want to ask, one of the interesting questions is, you know, if we plot these with respect to the real temperatures, do these lines cross? And if so, where do they cross? Right? At what temperature and what kind of ecosystem do they cross at? And I'm oversimplifying a lot here, but, you know, I think this is one of the, you know, the relatively straightforward ways to make a connection between this kind of theory, which I think is well rooted in biochemistry and physiology, you know, to a global ecological process. And the hypothesis is that where this crossover occurs is roughly at the transition between deciduous temperate forest and coniferous forest. And when we have environments that are colder than that transition, the rate of, this is inverse temperature, so when we have environments that are colder than that transition, the rate of photosynthesis exceeds the rate of respiration. And what does that mean? It means that those ecosystems store carbon. And that's what is stored as peat or muskeg or just unused carbon. If you go to the Olympic Peninsula, there is a huge layer of undecomposed carbon, everything from whole trunks of trees to great depths of litter in those relatively cool forests. In warmer environments, the rate of respiration could exceed the rate of photosynthesis. But of course, you can't respire what's not produced. So the implication here is that in these warmer environments, respiration is pretty well able to keep up with photosynthesis so that most of what is the carbon that is fixed is respired very rapidly. And if you go like to Steve Hubbell's tropical forests, there's not a big litter layer on the floor there, and if a trunk falls down, it decomposes pretty fast, right? So here we have rate of photosynthesis greater than respiration, and carbon is stored when it's cold. Here we have respiration greater than photosynthesis, and so CO2 is released and recycled at the same rate, roughly the same rate that it's fixed. So now what happens if we warm up the planet, right? What happens is that we shift the relationship up here, and this carbon that's been stored up is going to get respired. And here are the real data from these eddy flux towers, which show you whether and where these relationships may actually cross. So these are ones that Krista Anderson Teixeira and Marcy Litvak worked on around Albuquerque. So here's the rate of photosynthesis in blue. Here's the rate of respiration in red. And what we see is that it actually looks as if the crossover occurs in these New Mexico habitats at warmer temperatures than I suggested before. But the slopes are clearly different in the direction that you expect. And they do cross over. And there's some interesting other implications of this. In some of these New Mexico habitats where it's very cold, they don't seem to cross over at all. And of course, in many of these dry habitats, we have this additional phenomenon that a big source of oxidation of fixed carbon in the form of biomass is fires, right, which will periodically move through these communities. But still, the implication, if we sort of scan back out to the globe, is that in cold environments like boreal forests, the rate of photosynthesis is greater than the respiration. CO2 gets stored up in the litter. And if we warm those up sufficiently, we expect then to release carbon, which potentially provides a positive feedback into the global warming. And this is a phenomenon that the atmospheric scientists have become paid increasing attention to within the last few years, is this positive feedback from oxidation of stored carbon in these cold ecosystems as they're warmed. And the reverse here in tropical forests, where the fixed carbon is respired rapidly. So if we warm up a tropical forest just from this crude temperature dependence, what we predict is that we just simply increase the rate of carbon turnover somewhat. But there could be all other kinds of effects. Okay. I want to end, so the stuff I've talked about so far, I think, the theoretical foundations are pretty strong. And, we can argue if we want about whether we want to use this hard version or a softer version of a metabolic theory. But I think this metabolic way of thinking about ecological patterns, and particularly about the processes, the mechanistic processes that underlie them, is very powerful. I was talking to Malcolm Gordon before he came up here, and I was saying, I spent a large fraction of my career doing what we call physiological ecology, or environmental physiology, and I never kind of got this metabolic perspective, that ecology is about these exchanges, and to understand those exchanges and develop a theory for that, we ask the question, what flows, and what are the rules that govern those flows? And some of those rules are physical laws, like the laws of conservation of mass and energy and the laws of thermodynamics. Some of them are chemical laws, like the laws of stoichiometry, and some of them are biological laws, if you want to call them like these scaling relationships, how these biological rates vary with body size and temperature. So now I want to introduce you to something that I don't understand. And I'm going to admit right up front that I don't, and if you have questions or discussion about that, I would gladly entertain that. But the question is, can we move toward a metabolic theory of biodiversity? And because diversity is ultimately supported by these flows. And I'm going to simplify a lot here, and I'm going to leave out, with due apology to Victoria Sork and maybe the others of you, some of the other hypotheses that we might have for some of the controls on species diversity. But I think if you ask the question, what mechanisms have been suggested for ecological controls on species diversity, you'd get primarily two hypotheses. And the old one, and the one that I helped develop early in my career, and which I still think is part of the story, is that diversity is related to productivity. And the simple, dictum that characterizes this is that a larger pie can be divided into more species. If I have a more productive ecosystem, like a tropical forest, we can divide that up more finely among individuals and species, and have each species get enough so that it avoids extinction and remains in the system. And we can see this reflected potentially in the relationship between species diversity and body size. So small organisms require fewer resources than large ones, so we can divide the world up into a lot of small organisms and a smaller number of large ones. And we can see this with respect to temperature, because warmer environments are more productive, and so they're going to have higher rates of production. And I'm not going to say anything about stoichiometry, but it's obvious that there is a substantial effect of stoichiometry, that nutrients in water limit productivity. Diversity is low in deserts because water is limiting. But I don't want to get into that here. What I think is more interesting, and is maybe suggested, at least in my own mind, by this metabolic theory, is that there's an important role of kinetics in generating and maintaining diversity. And we've developed this little dictum then, that the Red Queen runs faster when she's hot, right? When we warm things up, everything goes faster, and that there's something, and I won't be able to be much more precise about that, there's something about everything going faster and interacting faster that generates and maintains biodiversity. And actually, if you think about it, some of this effect of body size could have to do with this kinetics, because small organisms have higher rates of turnover. They do things faster than larger ones. So the body size effect may not just be an effect of productivity. But then, we know, and we have good evidence, although I won't go into it now, that the rates of biotic interactions and pretty good evidence now, I think, that rates of evolution and speciation are temperature dependent. But still the question is, how do we go from this kind of phenomenology, this kind of vague qualitative phenomenology, to a more mechanistic understanding that we can maybe write down with some of these kinds of models. And you'll see that I'm not going to get there yet. So here's the body size effect. This is classic paper by Bob May back in the 1980s, and this is supposed to be the best estimate of the number of species of animals, terrestrial animals, as a function of their length here. And here, you have this increase. You have more small organisms than large ones. These are both log scales again, up to some size here. And May's question mark really refers to the question of whether this is due to undersampling and diversity, whether diversity just doesn't keep on going up here. And I think the more evidence we're getting on microbial diversity, the more likely it is that this just keeps on going, maybe even all the way to viruses. So there is a big effect of body size, and I'm not going to talk about that anymore, however. I want to talk about mainly the effect of temperature and these global gradients. So if we get these kinds of maps, and between GIS and various surveys that we're doing of different plant and animal groups, we're getting now these maps for most of the major taxa of organisms, both terrestrial and aquatic. If we do this for terrestrial organisms, we get a pattern like this. So this is global diversity of amphibian species. It's high in the lowland wet tropics, and it declines conspicuously with increasing latitude. It declines with increasing elevation, and it also declines as we go into arid areas like the great deserts of the world. And I won't quantify this for you, but we see this pattern over and over again in most plant and animal groups. So here's the Bonn group in Germany, species diversity in vascular plants, high diversity through here, declining as we go toward higher latitudes, declining as we go into the deserts. And it's interesting that we see this even in mammals, which take their body, which are endothermic, so they take their body temperatures with them wherever they go. But still, each dot is in the center of the geographic range of a mammal species. And you can see that they're clustered in the lowland wet tropics, and they decline with increasing latitude toward the poles and as we go into these arid regions. So over most groups, these patterns are very congruent. This is a very interesting one, I think, and I won't talk about this later this afternoon, but we've become quite interested in human ecology. And each dot here is in the center of an Aboriginal language group. And you can see that the correspondence with the previous is obviously extremely close, right? Different cultures are packed very closely into the lowland wet tropics. And this extends down, so here's the correspondence as a function of inverse temperature, again, this kind of Arrhenius plot. And this is diversity per kilometer squared. And the green dots are the mammal species. The red dots are human languages. And the correspondence is uncanny. And we can, again, talk about that. I don't have a great explanation, but I think that this is not coincidental, that this high degree of correspondence reflects something about the underlying processes. If we focus in a bit, here's the diversity of ant genera in Australia, which is high around this wet rim around the outside here, very low in the arid deserts in the center. And this is both warm and wet up here. So look at that. And this is Tyndale's map of territories of Aboriginal tribes in Australia in 1930 something, based on the best estimate sort of at European contact. And where are they most densely packed? In almost exactly the same, I'll skip back to the ants. Look at that, right? I mean, the correspondence is uncanny. All right, so then the question is why? If we look at primary productivity, in terrestrial environments, there's a high degree of correspondence between those patterns of biodiversity that I showed you and productivity. And I won't plot graphs there. And these highly productive areas are also, in general, the warm areas through the lowland tropics here. But we have a natural experiment that to some extent allows us to tease apart the effects of productivity versus temperature on diversity. Because if you go to the marine realm, there's no correspondence, there's no correlation between primary production and temperature. And in fact, some of the most productive marine waters are some of the coldest marine waters. And this is because productivity in the marine realm is controlled primarily by nutrient supply. And so, where is productivity high? It's where there's upwellings and at the outflows of major rivers. But if we ask where diversity is high in the oceans, we see that there's this dramatic signature of temperature. So these are coral reef animals, fish, corals themselves, and gastropod mollusks around the world. And in most of these benthic marine organisms, the center of biodiversity is this area of the Indo-West Pacific. And it's not just a matter of temperature, but obviously high diversity is in these warmer areas. Oops, if we look at pelagic marine fish, so fish out in the open waters, we get the same picture. High here in the Indo-West Pacific and in other warm waters, and very low in some of these very cold areas. So, kind of here's where I am in my thinking about this, that metabolic ecology should be able to explain, this is a premise, should be able to explain the major patterns of species diversity, and it should be able eventually to account for these patterns of diversity with respect to body size, latitude, elevation and depth, etc. And in general, now that we're getting good data, the patterns are clear. We see relationship with productivity, with temperature, with environmental heterogeneity, and now with the bow to Victoria and maybe others of you here, with phylogenetic and Earth history, but the underlying mechanisms are not. And I'm the first to admit that. So the question is, where do we go from here? And I'm going to leave that question unanswered. And so then, just to sort of sum up here, what's the role of either MTE with small letters, the gentle version, or the harder version? And I think especially the harder version, it has a number of things going for it. It's based on well understood first principles of biology, physics, and chemistry. It is admittedly, like any theory or set of models, a deliberate oversimplification of a more complicated reality. But one of the things it brings out is a fundamental unity of life that holds over all the living things, from microbes to monsters and molecules to ecosystems. Sometimes the slopes vary, sometimes the normalizations vary, but it provides a framework for unifying our understanding of life and how the fundamental characteristics of life at the cellular and molecular and biochemical level translates out to the ecological characteristics of organisms. It makes precise, quantitative, empirically testable predictions, and even when those predictions don't hold very well, they provide baselines for evaluating residual variation due to other factors. And I'm just going to skip over that, I think. So, as I said, I haven't done anything myself for a long while, I'm enormously indebted for everything I've said to a large number of collaborators, and one who I didn't list specifically here, but I think is somewhere in this room, is Van Savage. So thank you all. People need to go to lunch or something, go. I'll be happy to try to answer questions.
So, does the artifact form an example of this, or does it form a counterexample in terms of the pumping nature of resources?
Okay. I've used actually the species area relation for islands to argue that just increasing productivity itself is inadequate to account for the magnitude of the gradient that we see with respect to productivity and temperature. Because there's a very nice relationship, I think that's what you're referring to, between the area of an island and number of species. The slope of that relationship is about a quarter. You know, it can vary around a bit, and I don't want to make anything that there's anything magic about that quarter, but the slope is about a quarter. So that means that to increase diversity by an order of magnitude, tenfold, you've got to increase area by 4 orders of magnitude. And you know, if you increase the area 4 orders of magnitude, you get 4 times as much stuff there, roughly, presumably 4 times as much productivity, and you often have more environmental heterogeneity and other things that feed in to more species. But if you calculate the rate of increase in diversity in thermal gradients, either with latitude or with elevation, it's much steeper than that. And I can do a similar exercise. I didn't put it in here for time, but Steve Hubble and others have suggested, that one of the places we look to get some insight into the processes that underlie species diversity is the species abundance distribution in different environments. And I can make a similar argument there, suggesting that just increasing productivity and increasing the number of individuals, while it will increase diversity, it will not have the quantitative magnitude that we see even in these terrestrial environments. Yeah, Steve.
I just wanted to comment that there's some wonderful data coming out of Carlos Harmia's lab in Stry, looking at the pollen diversity going back to the Eocene, and the pollen diversity is absolutely perfect.
Yep.
It's just a beautiful trajectory through the tertiary.
Yep, great. I haven't seen that, but I'm delighted to hear it. Malcolm, yeah.
To follow up on what Steve just mentioned, have you looked at the long-term fossil record with respect to estimates of overall biodiversity in different periods as compared with the overall climate?
With the paleo temperatures.
Temperatures?
We have not. Yep. Yeah, we have not done that. We've done some other stuff on body size in the past and so forth. I mean, we ought to do it. There have been a couple of studies done sort of at a smaller scale with the kind of thing that Steve was talking about by other groups. Well, actually, one guy in our group on the increase in diversity over the last 30,000 years from across the Pleistocene to Holocene transition, in that episode of global warming, and the rates at which species diversity increased back at the higher latitudes, for example. And one of my students did a study on that, and David Curry and some of his people in Canada have done. And I think you're going to see that pattern. When you had warm conditions and tropical rainforests in Alaska in the Eocene, you had diverse forests there. I don't know if we know exactly how diverse the tree species were there, and so I'm delighted to see, hear Steve say that it looks like the quantitative patterns actually support this really well. Yep. Victoria.
Your front row is dominating right now. So two observations. When I saw the map with the linguistic map, what immediately pops in my mind is the fact that the restriction of migration. What you see in human population groups is when they're not migrating, it's going to maintain both linguistic and population genetic structure. So Now, in some ways, that plays into productivity,
Right.
because if you've got enough food locally, you're not going to be migrating around. Then that backed me into when I first saw your mammal, before I got to the human, I was thinking, when I see that, being the bias that I have is,
Right.
I see is that you have a lot of local population stability that allows species to stay separate, new ones species not to merge and stay separate. But there's a final comment, and then you can synthesize it. When I look at in ecological niche modeling, correlate that with haplotypes, in areas that have long-term stable niches where the populations are not shifting around a lot, you get a maintenance of high haplotypic diversity. And if I go to an area that's also been warmed but has a lot of shifting over time, a lot of information, you get shifting of climate, what you do is you lose. Presumably, you're losing through genetic drift.
Right.
So those are three
Right. So I think there's a really interesting, this is, I think, a case where the humans may actually help more clearly about the biodiversity patterns. Because one of the big hypotheses that's on the table that I didn't even mention explicitly is this idea of niche conservatism, the idea that species originate in the tropics and they tend to have conservative niches and they require special adaptations to get out. And just most of them don't manage to do that, right? I mean, this is greatly oversimplifying, right? But that cannot explain this pattern of diversity in the New World. Humans came into the New World from up here, somewhere on the order of 10 to 15,000 years ago, and they spread down here, and they independently produced these high-density cultures with lots of linguistic diversity. They independently evolved agriculture and complex societies, and so forth and so on. So now, they may have independently acquired genetic structures of the sort you're suggesting, and I certainly wouldn't want to rule that out as part of the explanation. Yeah.
Jane.
We usually look, ecology look at body time. You see time as long as you're looking at the earth organism over fairly small elevation radius. But I wonder, what happened here at the center of the oxygen? That's important for aquatic organisms, important, and also important over geological time, where oxygen concentration have been rising. So I'm wondering if anyone would try to even develop a theory of what has to be affected as oxygen is.
Yeah, I know only enough about that to probably get myself in trouble really fast. Oxygen is, in that sense, comes in, I think, as another one of these stoichiometric variables. And clearly, there are lots of areas, there are deep water areas that are anaerobic or have very low oxygen and very low diversity then. Aerobic organisms basically can't live there. So that's another, that's sort of the water, I view that kind of maybe as the water equivalent of, very severe stoichiometric limitation. Whether, how well diversity sort of scales out on a gradient of oxygen availability, I don't know. And of course, that's complicated by the temperature effect because you can, cold water can carry more dissolved oxygen than warm water. So there are a bunch of complications there. But yeah, once you get to aquatic environments, oxygen limitation can play obviously an enormously important role. No argument.
Any other questions?
No, I'm going to see you at 3 hours.
Yeah, we got a lot to talk about. We won't get it all done, but
Going twice. Please come at 4 o'clock for his next talk, which will
Yeah.
link ecology with humans.
hopefully